How many times have you been told that you have your mom's eyes or your dad's nose? If we look back in our discussion on the cell cycle and cell division, we have established that our traits are combinations of traits from our parents. Because of the process of crossing over during prophase 1, new combinations of traits are made, which is why we are not perfectly identical to one of our parents. These patterns of inheritance are best studied in the field of biology known as genetics. Genetics deals with heredity, or the phenomenon in which traits from parents are passed on to their offspring. Centuries ago, scientists believed that the traits of our parents are blended together as they produce offspring. For example, if your mother has curly hair and your father has straight hair, you will have wavy hair. They called this the blending hypothesis. However, a monk named Gregor Mendel disagreed with this hypothesis, as his experiments on pea plants suggested that these traits are inherited directly and not blended with each other. In the same example, if your mother has curly hair and your father has straight hair, you will either get curly or black hair. This was called the particulate hypothesis. This eventually gave rise to what is now called Mendelian genetics. So who is Gregor Mendel? Gregor Mendel is the abbot of a monastery, and during his free time, aside from praying, he liked to garden and do math. While planting garden peas, he noticed that there are certain patterns that appeared in the offspring every time he would plant a new generation of peas. The first thing that he observed was that the garden piece has distinct traits, and these traits were also in twos. For instance, the flower color is either purple or white, and the seed color yellow or green. He also noticed that he can control the mating of these peas. These plants can reproduce by itself, and they can also cross-fertilize, which is done by mating one pea plant with another pea plant. Since he had a lot of free time, he decided to perform some experiments. So he took one purebred purple-flowered pea plant and he mated it with another purebred purple-flowered pea plant. When you say purebred, the parents of the plants were also purple-colored, and the parents of those parent plants were purple as well. As expected, crossbreeding a purebred purple-flowered pea plant would create offspring that are all purple-colored. So he decided to take this one step further. Mendel decided to mate a purebred purple-flowered plant and a purebred white-flowered plant. These two comprise the parental or the pea generation. If the blending hypothesis is true, then the next generation or the F1 generation would be a blend of purple and white and will produce light purple colored flowers. To his surprise, all of the offspring after this cross were purple. The big question is, where did the white flower color go? Taking it another step further, he decided to crossbreed two members of the F1 generation, all with purple flowers. To his surprise again, most of the second generation or the F2 generation had purple flowers, but some had white flowers. He then hypothesized that one of the two traits is stronger or is more dominant than the other, to the point that if these two versions of the same trait is found on the same plant, then the more dominant trait will mask the expression of the weaker or recessive trait. Mendel observed the same phenomenon when he studied the other traits of the pea plants, such as seed color, seed shape, stem length, etc. If we take Mendel's findings in the modern context, we know that each diploid cell has two copies of a chromosome, one from the mother's egg and the other from the father's sperm. These chromosomes are homologous or have the same traits. These similar traits are called alleles and are found in specific locations or loci in a chromosome. In the slide, you can see that the allele for purple flower color and the allele for white flower color are found on the same region of the two chromosome copies. Mendel concluded that for each trait, we inherit two alleles, one from each parent. If the two alleles are carrying the same version of the trait, then the alleles are called homozygous. For example, both chromosomes are carrying the trait for purple flower color. If the alleles are carrying different versions of the trait, for example, one copy is carrying a white flower color and the other a purple flower color, then they are referred to as heterozygous. If the alleles are heterozygous, one of them is more dominant than the other, and this more dominant allele will determine the appearance of the trait, while the recessive allele counterpart will be masked and will have no effect on the appearance of the organism. This tells us that our traits do not necessarily tell us our genetic composition. A flower can appear purple, but its chromosomes might have the allele for purple flower color and the other allele for white flower color. The alleles that we possess for each trait is called the genotype, while the appearance that our traits exhibit is called the phenotype. One systematic way to predict both the genotype and phenotype of crossbred organisms with known genetic makeup was developed by Reginald Punnett and is appropriately called Punnett square. In a Punnett square, a dominant trait is represented by an uppercase letter, while a recessive trait is represented by a lowercase letter. Let us try to analyze Gregor Mendel's observations using the Punnett square. Let us use the letter P to represent the alleles, with a capital P as the dominant trait, in this case purple flower color, and the recessive trait with a small p, the white flower color. The traits are arranged this way in the Punnett square and are then crossed. By crossing the alleles of one parent with the other, you are able to come up with new allele combinations. In this case, you can see that there are 4 out of 4 chances that the offspring will have a genotype composed of a capital P and a small letter p. Since purple is a more dominant trait over white, then all of the offspring will have purple-colored flowers as their phenotype. This explains why in Mendel's cross, the white flower color suddenly disappeared in F1. Mendel then crossbred 2 members from the F1 generation, each one with a heterozygous genotype with a big T and a small p. In the resulting F2 generation, different results were observed. For the genotype, you could see that there is a 1 out of 4 or 25% chance that the offspring will have a genotype that is homozygous dominant, or the two alleles being big P and big P. There is also a 2 of 4 or a 50% chance that the alleles will be heterozygous, one big P and one small p. And lastly, there is a 25% chance that the alleles of the offspring are homozygous recessive, that is, 2 small p's. When translated to the corresponding phenotype, there is a 75% chance that the trait will appear purple, and a 25% chance that it will appear white. The appearance of the white trait can be attributed to the homozygous recessive genotype. Because there is no dominant purple trait present to mask it, the recessive trait can then be observed. Let's try some practice problems. The ability to taste a bitter chemical, phenylthiocarbamide or PTC, is due to a dominant gene. Use capital T and small t to symbolize the 2 alleles of this gene. For the first question, what is the genotype of a non-taster? What are the possible genotypes of a taster? The answer, of course, is for a non-taster, it should be 2 small t's, while for a taster, it should be either 2 big T's or 1 big T and 1 small t. Now for the second question. If your mom is a heterozygous taster and your dad is a homozygous non-taster, what are the chances that you are a taster? To approach the problem, first determine the genotypes of your parents. Now perform a Punnett square. Based on the results of the Punnett square, These are the genotypes and phenotypes. This means that there is a 50% chance that you are a PTC or a phenylthiocarbamide taster. How about this problem? Polydactyly, or having more than 5 fingers and or toes, is a dominant trait. A woman, homozygous for polydactyly, is married to a normal man. What are the chances of producing an offspring that has extra fingers and or toes? Use the letters capital P and small p to represent the alleles. Pause the video right now so you can try solving for the answer. Based on the Punnett square on the right, there is a 100% chance that their offspring has an extra finger or an extra toe. Here's another problem. Albinism, the total lack of pigment, is due to a recessive gene. A man and a woman plan to marry and wish to know the chances of them having any albino children. What are the chances if the man is an albino, the girl is normal, but her father is an albino? Use the letters capital A and small a to represent the alleles. Again, pause the video right now so you can try solving for the answer. Now this might be a little tricky. The first step is to find out the alleles of the parents. Since albinism is a recessive trait, the father's alleles should be 2 small a's. The mother's alleles, on the other hand, are heterozygous. How do we know this? We know that her father is an albino, so his alleles are 2 small a's. This means that one of her daughter's alleles should be a small a. Since she appears normal, this can only mean that she has a copy of the dominant allele, big A, that masked the albino allele from her father. By performing the Punnett square, you should come up with the following genotypes and phenotypes. The answer to the question would be, there is a 50% chance that their child will be an albino. Punnett squares are applicable to many of our traits as well, so one reason to learn it would be this. To summarize, these are the principles of inheritance established by Mendel encapsulated in Mendelian genetics. We have learned that genes are sequences of DNA that codes for a trait. Alleles are versions of one gene, and a homozygous trait means that it has identical alleles of a gene, while a heterozygous trait has different alleles of the gene. Genotypes are the sets of alleles, while the phenotypes are the corresponding observable characteristics. Alleles can also be dominant, which can mask its accompanying allele, or recessive, which is masked by its accompanying allele.